In part 1 of the DNA lesson, we learned how DNA can be duplicated for the purpose of cell reproduction. In this lesson, we'll look at how cells can use the information stored in DNA. They use the information in DNA during interphase of the cell cycle, where the cell is growing, metabolizing sugar, and importantly, producing proteins. Now, we've learned that proteins are the workers of the cell. They perform the chemistry of life. Some of that chemistry is breaking down food molecules to get energy. Other chemistry involves building important cell molecules. So proteins are the important workers of the cell. They keep the cell alive. So it stood to reason that if DNA was storing information of some kind, it would probably be the information to build proteins. So somehow the DNA molecule has recipes to build proteins. A clue to how that might be the case can be seen if we take a closer look at proteins. Over here on the right, we see a protein. Now, all proteins have a certain shape, and it turns out they can be unfolded into a chain of chemicals called amino acids. So proteins are chains of amino acids. Some proteins are just several amino acids long, some are hundreds of amino acids long. There are 20 different kinds of amino acids that all proteins are composed of. Now, if we take this protein here that is attacking glucose, it might have a certain sequence of amino acids as shown here. But a different protein, perhaps the green one here or the orange one here, would have a different sequence of amino acids. This is significant because the sequence of amino acids determines how the protein folds up into a shape, and the shape determines its function. So a protein with a different amino acid sequence will fold up differently and therefore perform a different function. And this is why we drew those proteins with different shapes. Their shape is important in their function. So shape determines the function, and the sequence of amino acids determines its shape. So back to DNA then. Somehow this sequence of DNA letters specifies how to construct a certain given protein. And what scientists concluded then is that the DNA sequence along one strand is coding for another sequence of chemicals, this time the amino acid sequence of a protein. Modern biologists now understand that it is a sequence of 3 DNA letters along one strand of a gene that codes for a given amino acid. So the first 3 letters in the DNA sequence stands for or names the identity of the first amino acid in a protein. The second 3 letters is the identity of the second amino acid. The third 3 letters is the identity of the third, and so on. So in this way, then, a sequence of letters that appears to us to be random, to the cell has meaning. Every 3 letters of DNA is a name for one of the 20 different amino acids. Now, as it turns out, there are 64 possible DNA triplets, right? So the first position of a triplet, there could be any one of 4 letters here, an A, a G, C, or T, and same with the second and the third. So there are 64 possible DNA triplets, but only 20 amino acids. This means there's redundancy in the genetic code. For example, right here, the amino acid here, the red square, has been repeated here. So the second and third amino acid is the same amino acid, but the triplet is different. So the redundancy means that each amino acid, each of the 20 amino acids, can have several triplets that specify that amino acid. Now in this diagram we see a cartoon amoeba, and we're going to use this drawing here to illustrate how cells use DNA information to build proteins. The proteins are going to be the ones involved in pseudopod control, so we have receptor proteins, microtubules, and choppers. Let's go into the nucleus to begin. So in the nucleus we have the green molecule. The double helix here would be DNA. Some of it is wrapped around a histone protein here. Of course, any information that's wrapped up cannot be accessible, so you can't make messenger RNA from DNA regions that are wrapped up. But here we have 3 genes that have been opened up. Remember, gene refers to the information content. DNA is the actual molecule that has the information. So here we have the DNA has been opened up, the strands have been separated, and messenger RNA is being constructed. Here we have a different gene, a different recipe for a different protein. The blue messenger RNA is being constructed from the bottom strand of DNA. And here we have a third gene. Again, the DNA strands have opened up, and messenger RNA, the black one here, is being built from the bottom strand of DNA. Now what will happen then is that these messenger RNAs will leave the nucleus and hook up to the factories called ribosomes. So the red one is shown over here. Now, this gene is the recipe to build the receptor protein. So as the ribosome goes down the length of the messenger RNA, it's reading the RNA triplets 3 at a time, and it's linking amino acids in the order specified by the triplets of messenger RNA for this particular protein. Since the microtubule is a different protein, the messenger RNA triplets are going to be different, and therefore the sequence of amino acids will be different. And again, the chopper protein will have a different messenger RNA sequence, so the amino acid sequence will be different. And because the amino acid sequences are different, the proteins fold up into different shapes. Because they fold up into different shapes, they perform different functions. Let's take a closer look what was happening there in the nucleus. In this picture here, we see our gene. So again, the information content, that's the recipe for a given protein, and then the double helix molecule, that's DNA. The DNA strands have been separated, but this is not for the purpose of DNA duplication. This is to make messenger RNA, the first step in building a protein. So in the nucleus are spare RNA letters. Notice the difference. One difference is that RNA has a U, whereas DNA has a T. But if you'll look at the green RNA molecule here, you'll see that the pairing rules apply. It's just that whenever there's an A in DNA, there's a U in RNA. Now, another protein, not DNA polymerase, but RNA polymerase, is going to be responsible for chugging down the strand of DNA, building messenger RNA by linking the RNA letters together into a single-stranded, stable RNA molecule. So RNA polymerase is doing a similar kind of job as DNA polymerase. It's just RNA polymerase is making messenger RNA, whereas DNA polymerase is duplicating DNA. When the messenger RNA molecule is finished, in other words, when RNA polymerase gets to the end of the gene, the RNA molecule will detach from the DNA and leave the nucleus, heading out into the cytoplasm to find a ribosome. At that point, the two DNA strands close up again, and they just kind of stick to each other and then can be coiled up again, or can be twisted again into the double helix. Now let's take a look at what happens in the cytoplasm. The big green blob here is the ribosome, and you'll see it's hooked up to a messenger RNA molecule here. Notice the sequence of letters. Now the ribosome's job is to read the messenger RNA 3 letters at a time and then link amino acids in the order specified by those triplets. Now we're not going to get into the details, but these tRNAs, these are transfer RNA molecules. These are the ones that bring the amino acids to the site of construction. And they happen to have letters themselves that stick to the triplet on the messenger RNA, and that's how the amino acids can be properly positioned in order to be chemically linked together into a chain. But all we'll need to know for our purposes is that the ribosome's job is to construct a protein by linking amino acids in a chain, but the order of those amino acids is being specified by the order of triplets in the messenger RNA molecule. On the left here, we see the genetic code, and this table allows us to translate the language of messenger RNA into the language of amino acids, as shown on the outer part of the diagram. Now, let's do some practice. The first triplet here is CGU. So we start in the first position on the inner ring. It's a C, so we go to the C quadrant. Then we go, the second letter is a G, so we go to the G section, and third letter is a U, so within the G section we go to the U, and that's arginine. First amino acid is arginine. Next is GAC. We start with G section in the inner ring, A, and then C. That's aspartate. Third is AGU. AGU. That's serine. In fact, because the transfer RNA molecules here, this amino acid is serine. So it's being positioned now to join the growing chain of amino acids that makes up a protein. Let's find out what the red amino acid is. That's a CGU. So we have CGU. That's another arginine. So that was a duplicate there. The next one, ACG. ACG, that's threonine. And so the purple one is threonine. Let's find out what the green one was. Well, that was this triplet down here, UCA. UCA, that's serine again, and one more, the orange one over here, that's going to be GAU. GAU is another aspartate. Now, at the end of this messenger RNA will be a triplet. The black dots here are stop triplets. This is punctuation. These triplets do not code for an amino acid. They signal the ribosome that the message is complete, and the protein will detach and then fold up and go do its job in the cell. One more thing to mention about the ribosome and the messenger RNA is that when the protein is finished, the messenger RNA will detach from the ribosome. And it can then float around and bind to another ribosome and get translated again. And that will happen for some amount of time. In this picture here, notice we have the messenger RNAs leaving the nucleus, hooking up to the factories, but these are Hammerling's molecules, and we learned previously that messenger RNA molecules are temporary molecules. So they can be used for a while to make many proteins, but after a while the messenger RNA molecule itself will degrade. Now, it's interesting to compare messenger RNA with DNA. Remember, messenger RNA is a single-stranded molecule, so it's more vulnerable to destruction. The DNA molecule is a double-stranded helix. It's a more stable molecule, and it resists degradation. So the messenger RNA molecule will hook up to ribosomes, and it can be used many times. But in time, the messenger RNA molecule will degrade, and so the cell will have to make new messenger RNA molecules if it wants to keep making that same kind of protein. So again, the basic idea is that what the ribosome is doing is translating the language of messenger RNA sequences into proteins, which are amino acid sequences. And we do want to think of this as a translation. And this is different from what happens in the nucleus of the cell. So let's compare what's going on in the nucleus to what's happening out at the ribosomes in the cytoplasm. We're going to give two words for this. In the nucleus, we're going to call this transcription, and in the cytoplasm at the ribosome, we're going to call that translation. First, in the nucleus, we're going to call this transcription because to transcribe something is to sort of make a copy of it. Well, in the nucleus, remember the gene is opened up, the messenger RNA is being built from one strand of DNA, so it's sticky to this strand of DNA, but that means it's a copy of the sequence of this strand of DNA. So what the cell is doing is, in a sense, transcribing a gene into messenger RNA. It's making a copy of the information in DNA and building it into a messenger RNA molecule. So we call that transcription. Now, if we think about transcription in another context, imagine an author like Charles Darwin who wrote The Origin of Species. Let's say he wrote this book. Well, what if he were to sit down one afternoon and simply just copy his book twice? That would be transcription. So the cell is transcribing the information of DNA into a messenger RNA molecule. By contrast, out in the cytoplasm at the ribosomes are doing an act of translation. And that is, they are converting the language of messenger RNA into the language of proteins, which are amino acids. So these are two different languages. So that's a translation. It would be like Charles Darwin taking his original book in English and translating it into German. Again, translation involves taking one language and making a corresponding sort of mapping onto another language. In this case, the language of RNA triplets is being turned into the language of amino acids linked together to make a protein. So this is how cells use DNA information. They transcribe the information into a molecule of messenger RNA, which leaves the nucleus, and that's Hammerling's molecule. It hooks up to a ribosome, and the ribosome will translate that sequence of RNA letters into a sequence of amino acids, which is a protein.